Hello, everyone, and welcome to this Cell and Gene Live. As Heather said, I'm Erin Harris, and I'm happy you're here. Thanks for your time. As always, we have a terrific presentation lined up for you. I'm thrilled to have our expert panelists joining me today. They include Pratik Jaluria, Ph.D., SVP Technical Development Strategy at BlueRock Therapeutics, Bruno Marques, Ph.D., VP Process and Product Development at Century Therapeutics, and Kate Rochlin, Ph.D., COO at IN8Bio. I'd also like to thank Charter Medical for partnering with us on this event. We will be taking your questions, as Heather said, during the presentation, so please go ahead and submit them as you think of them via the Q&A tool. We love to hear from our audience, so please ask away. And before we get started, I'm going to have each of our panelists briefly describe their role and their company, which will help frame the POVs they'll share today. So, Kate, I'd love to start with you, if you could start us off.
Sure. Great to be here. I'm Kate Rochlin. I'm the Chief Operating Officer at IN8Bio. I've been there for about 5 years and lucky enough there to oversee our CMC, clinical manufacturing, clinical translational analytics, quality, and most of our technical side operations. We work primarily in gamma delta T cells and oncology. We are phase 1, phase 2 clinical programs, and we also have a preclinical program that's transitioning towards clinical in the iPSC gamma delta T cell space. So very excited to share my views with you on that particular brand of iPSCs, the gamma deltas, why we think they're so powerful, and all the excitement around still using them clinically. Great to be here. Thanks, Erin.
Yeah, thank you, Kate. And I'm really excited to hear from everything that you're doing and all that IN8Bio has to share with us. Bruno, next up, Century. Talk to us about it.
Hi, everyone. Happy to be here as well. So Bruno Marques, I lead the process and product development team at Century Therapeutics. And we are developing iPSC-derived immune effector cells to treat oncology and autoimmune patients. So I've been lucky enough in the 5 years I've been at Century to be able to work with a gamut of iPSC-derived immune effector cells, NK cells, gamma delta T cells, and now most recently alpha beta T cells. So happy to share some of that experience with you.
Thanks, Bruno. Appreciate it. Pratik.
Yeah, hi, Pratik. Happy to be part of this conversation. I'm part of the technical operations group organization at BlueRock. At BlueRock, we're a clinical stage company. We're focused on developing PSCs for a variety of indications, including neurological, ophthalmic, and our main focus is on developing what we believe are transformative therapies for patients.
Well, thank you so much. Pratik, while you're still in the hot seat, I'd love to start off with you. So organizations are always, looking for ways for, to more effectively bridge the gap between laboratory development and CGMP manufacturing, which will ultimately accelerate the path to clinical trials and commercialization of cell and gene therapy. So if you could talk us through, any of the best practices for translating what is done in the lab into what can be expected in a CGMP setting?
Yeah, great question. So the way that I think about it, or at least the way we think about it at BlueRock, is that, as I think many people in the audience can certainly appreciate, when you're doing activities in a development setting, you're doing them at a small scale, lab scale. And so translating that into what that might look like in terms of the number of cells that you're working with or the number of vials that you have requires some thinking about at-scale production, at-scale runs to help confirm what you're doing at the lab scale. So some best practices that might include things like stress testing. So if you're developing a formulation, cryopreservation formulation, or other elements, you want to be able to understand how those formulations play. So you want to understand the physiochemical characteristics of them, things like pH, temperature, but also if you experience some type of fluctuation in temperature, excursions, what does that look like? Maybe there's freeze-thaw cycles that you want to explore. And also then thinking about the raw material grades that you're using. So it's very common in a development setting to use research use only grade materials. What does that look like in a GMP setting? So you want to use the highest grade possible, typically multi-compendial if you can, or USP, EP grades. And you want to make sure that you understand how those raw materials and those excipients are playing into your actual impact on product. So those are some of the best practices that I can think of that are relevant, that people should really consider as they're taking and translating lab scale activities into what would be applied in a GMP setting.
Excellent. Thank you so much. Kate, I'd love to hear from you. Same question, best practices from lab to CGMP setting, what's IN8bio's or, just from a broad perspective, what are your thoughts on that?
Yeah, I think so much of what was just said resonates with me. I think one of the things that we've always seen is that lab scale behaviors of cells and cell expansion can behave very, very differently than clinical scale or large scale GMP expansion. And so I think that translating early when you feel that you're ready to go is really critical, because that scale up to what is going to be your batch size or what your aspirational batch size may be is really important to get right. And so I think understanding those cell behaviors in that environment as early as you possibly can is really critical. I also think, to the note about reagents, that's also very important, and making sure that you are using things, if you're using research use only reagents, to ensure that there are reagent grades that would be appropriate for the GMP setting, and understanding what the differences in those reagents actually are. Are there excipients? Are there different contaminants that may be in the research ones? Is there no GMP equivalent? So you need to know that early and make sure that you are using things that can translate. And the other thing I will note is that in some cases, depending on what systems you're using, there are licenses, there are costs that are associated with going into GMP, and knowing early if you need to negotiate any of those. Those can take some time, especially if they're contractual. So making sure you don't get to a point in your development where you feel you're ready to go, and then all of a sudden you're held up by sort of the paperwork contracting end of things. So I think those are really important considerations to take in early on, and make sure that you do that landscape view and feel confident that the process you're using will translate, so you don't get 90% of the way ready to translate and then have to backtrack and sort of redo a lot of the work with either different grades, different companies, or whatever it is.
For sure. We hear that all the time, certainly on cell and gene, that doing that upfront work is really important. Bruno, I'd love to hear from you too about your take on this question, and perhaps Century Therapeutics perspective.
Yeah, no, great points raised so far. I think that what I would add comes from a perspective, especially thinking about this very important specific issue of cryoformulation and drug product development, right? And maybe just getting into a little bit of detail here, at least at Century, the holy grail around cryoformulation, drug product development is really around trying to maintain the functionality of these post-cryo, post-thaw cells as close to the functionality of fresh cells that we can, right? And the more we learn, the more anxiety there is around that, because we work so hard to produce these cells, right, at the end of these fairly long manufacturing processes, and then we have this extra step that could radically change, right, the quality and the behavior of these cells. And how do we avoid that? And so because of that little bit of anxiety, I think that there is a temptation to make decisions as early as possible, right? So in a research setting, you think that you've done everything that you can to understand the mechanistic behavior of these cells and you're ready to go. But cryo specifically depends a lot on engineering controls of your process, right? So I'm talking about some very detailed studies around hold times, right? Around your wash systems, around how do you control your freezing rate. And those things have a huge impact on the final product, right? The post-cryo product. And a lot of research labs just don't have the equipment and the expertise to do that, right? So I think that finding the right time and the right part of the organization to run those studies before jumping to conclusions is really important. So that's the perspective I would add here.